Hi everyone, I'm Dr. Karen Mate and I work in the School of Biomedical Science and Pharmacy at the University of Newcastle. It's really interesting coming to work every day because I do lots of different things. My main responsibility is to teach university students about how the human body works. This is a really important job because these students are going to become doctors or nurses or pharmacists or physiotherapists. So they need to understand how the human body works so they can treat their future patients in the most effective way. Other students that I teach are learning to become medical researchers so they can come up with treatments and cures for new diseases like the coronavirus, as well as older diseases that have been around for a long time, like asthma or diabetes or cancer. Some of my classes have more than 500 students, so I'm kept pretty busy. When the students are on holidays, I still come to work and do some medical research on how to improve the health of older people, like your grandparents, so they can stay fit and healthy for as long as possible. When I was growing up, I wasn't what you might think of as a typical young scientist, although I did enjoy science and maths classes at school. In my spare time, I really enjoyed being creative and making things. My mother didn't have the opportunity to get a good education, but she was very creative, and it was Mum who introduced me to crafts like knitting, sewing and crochet. I picked up the basics pretty quickly, and then I enjoyed working on more and more complex and detailed projects. A lot of my projects were based on creating repetitive patterns, something that I've always found very visually appealing and satisfying. At first I followed instructions from books and magazines, but then I started making my own designs. When I started looking around for inspiration, I started seeing beautiful patterns in nature, in the arrangement of leaves, on plants, the patterns on animals, like on snakes and giraffes and tigers, and I started incorporating these into my projects. One of the favourite things I made was a zebra pattern jumper that I wore all the time until I lost it. I haven't thought about that jumper for a while, but I wish I still had it. Maybe I should make another one. Those were some of the things that I loved doing as a kid, but I also wanted to share something about what I hated doing, and that was sport. I hate it because I was bad at it. Actually, I wasn't just bad, I was terrible. When we had running races at school, everyone else had finished before I even made it to the halfway point. I always dropped the ball in ball games and could never hit the ball in softball or cricket. No matter how much I tried, I couldn't get any better. My whole family was the same. We were all hopeless at sport. Have you ever noticed that some things run in families? Maybe everyone in the family is blonde, or good singers, or tall, or maybe they're hopeless at sport, like at my family. Anyway, being bad at sport meant that I spent more time doing other things that I did enjoy, including my schoolwork and craft activities. As I learnt more about biology, I found that there were beautiful patterns in the structures that make up our bodies. Our bones, cells, and even tiny molecules are arranged in beautiful ways, and these arrangements are important for the way that they work. You might have noticed the model that's sitting next to me. It's a molecule called deoxyribonucleic acid, or DNA for short. DNA is a beautifully intricate molecule that works in complicated ways to control what's happening in every cell of your body. If we look a little closer, we can see that it's got some interesting repeating patterns in its structure. First, notice its spiral shape. If it was bigger, it would look like a spiral staircase. Running along the outside spiral of DNA is what we call the backbone of the molecule. So it's made up of these repeating purple and red units that are really important in holding the structure of the molecule together and keeping it stable. The red bits are a kind of sugar, whereas the purple bits are another kind of chemical called phosphates. and they link together to form this sugar phosphate backbone along the outside spiral of the DNA molecule. The inside part of the DNA is made up of little rungs, like the rungs of a ladder. The rungs are really special, and each one is made up of 2 parts or bases. Each base has a name, but they're usually called by their initials: A, T, C, and G. In this model, A is orange, T is blue, C is green, and G is yellow. They don't like to be by themselves, so they always pair up with a partner, but they're very choosy about their partners. A and T, or orange and blue, always pair up together. And G and C, yellow and green, they always pair up together as well. In the body, DNA is a really long molecule that's coiled up like a noodle inside every cell. It's the order of the A and T and C and G bases along the length of that DNA noodle that controls everything that is happening in every cell in your body. That's a very big job. Our bodies are made up of about 30 trillion cells. That's 30 million, or a 30 with 12 zeros after it. That's a big number. If we just think about one of our hands, they've got around 2.5 billion cells. That's 2,500 million cells, but they're really tiny. So tiny that we can't see them. If we increased every cell in your hand just to the size of a grain of sand, your hand would be the size of a bus. So even though they're very small, every cell in our body has got its own job, just like humans do. Some cells help us to detect light and see. Others help us touch. Others help us hear. Other cells carry oxygen around our body. Other cells help us to digest food. There's more than 200 different types of cells in the body. So that's 200 different jobs that need the right tools and equipments to happen. But how does every cell know what job it has to do? Our cells are told what to do by this very special molecule called DNA. DNA is a record of instructions telling the cell what job it's going to be doing. It's written in a special alphabet that's only 4 letters long. It's those 4 bases that we saw in the DNA model: A, T, C, and G. But how could all of the instructions needed by all of the cells in our body be spelt out using just 4 letters? Let's get out some Lego and take a look. In this box, I've got 4 different Lego colours. I've got pink, I've got blue, I've got white, and I've got beige or light brown. So let's build a Lego tower 10 blocks tall using only those 4 colours. How many different combinations do you think you can make? Well, for the first block, I could choose any of these 4 colours. I might choose blue. And then for the next block, I've got another choice of 4. I could choose another blue one, or I could choose pink or white or beige. Let's go with beige this time. And then for the third one, I've got another 4 choices to make. Again, do I go beige, blue, white, or pink? Let's go for another blue. Okay, do you know that when I get to 10 blocks tall, there's 1 million different towers that I could make using just those 4 colours? So that is how DNA can store so much information with just 4 letters in its alphabet. And DNA is also a lot longer than 10 blocks, so it gives a lot more than 1 million options. There's more than 5 meters of DNA in every one of our cells. If we took all of the DNA in our body and stretched it out end to end, it would reach from the Earth to the Sun 100 times. There's so much DNA in our cells that we can do an experiment at home or at school and collect enough of our own DNA to be able to see it. Let's head to my kitchen and I'll show you how. This is what you're going to need to make your DNA. A couple of clear plastic or glass cups, some dishwashing detergent, preferably colourless, some measuring spoons, a 1/4 cup measure, some salt, a couple of drops of food colouring, some isopropyl alcohol or rubbing alcohol. You can get that at the chemist or at car shops, and 2 cups of drinking water. Okay, are we ready to make some DNA? The first thing we need is 2 cups of water. We're going to add a tablespoon of salt. Then we're going to take a 1/4 cup of that salt water and pour it into one of our plastic drinking cups. The next step then is to swish that salt water around in your mouth for 1 whole minute. Okay, so what we've done by swirling that salt water around is to collect some cells from the inside of our mouth. The next step is to add just a couple of drops of dishwashing detergent. That's going to break open those cells, and the cells are going to release their DNA. Try not to get too many bubbles. The next step then is to take a 1/4 cup of the rubbing alcohol, put it into our other clear cup, and add just a drop of food colouring. That's going to make it a little bit easier to see the DNA. Swirl that around, and now we are going to gently pour that alcohol on top of the salt water that we had swirling around in our mouths. So pour it gently on top, and you'll see that it forms 2 layers. The DNA is going to form at the interface or in between those 2 layers. So it usually takes a few minutes, so we just need to be patient and wait. It's been a couple of minutes since we put the alcohol in, and we can see little fine strands of DNA floating around in the cup. I'm going to try winding it onto my chopstick, see if I can collect it. Oh, look at all that DNA. Okay, so we've managed to isolate a large amount of DNA out of my cheek cells. Look how long it is. Okay, so I hope you have the chance to try that experiment at home or at school and are able to isolate your own DNA. Okay, bye for now.